So good evening. Today is the ninth problem solving session for the course Overview and Integration of Cellular Metabolism. The course instructors are Professor Arithri Veer and Professor Arindam Ghosh from IIT Kharagpur. And I'm Shrija Ghosh, a PMRF from IIT Kanpur. I'm here to help you solve your assignment questions related to this week's topics. So this week we'll be discussing metabolism of different amino acids. So last week we had discussed tryptophan, tyrosine, and phenylalanine. Basically, the aromatic amino acids. Today we'll discuss the rest of the amino acid metabolism and the associated disorders. So I've prepared certain questions that will cover all those topics. So feel free to ask any doubt in any of the questions, or if you have any doubt in any of the topics also, let me know. I'll try to help you. Okay, so I'll start with the questions now. The first question, which amino acid is the main methyl group donor in transmethylation reaction? Which amino acid is the main methyl group donor in transmethylation reaction? Option A, methionine. Option B, serine. Option C, glycine. And option D, threonine. You can write the answer in the chat box. Which amino acid is the main methyl group donor in transmethylation reaction? Methionine, serine, glycine, and threonine. You have answered option A, methionine. That is the correct answer. So methionine basically gets activated in the form of S-adenosyl methionine. So you can see this is the methionine structure. It has one sulfur group, and this sulfur group basically gets attached to the adenosine part. And from where the adenosine comes from? Adenosine comes from ATP. So you know ATP is adenosine triphosphate, right? So from that, the adenosine will get attached to the sulfur, and the product that we'll get will be S-adenosyl methionine. And this is a activated form of methionine, and the methyl group here shown in pink, it is it can be easily donated to different variety of substrates. So we'll see there are different methylation reaction where S-adenosine methionine acts as a methyl group donor. So it can give this CH3 group to any of the substrate, and in turn it will get converted into S-adenosyl homocysteine. Okay, and this is just the methionine metabolic pathway where methionine is converted into S-adenosyl methionine, and S-adenosyl methionine, once it donates the methyl group, it will become S-adenosyl homocysteine, which will later get converted into homocysteine by hydrolysis. And then from the homocysteine, it can again get converted into methionine by methionine synthase. Okay. So methionine gets activated by taking up an adenosine molecule from ATP and become S-adenosyl methionine, which can donate the methyl group easily to any substrate. Moving on to the next question. S-adenosyl methionine is a methyl donor in all the reactions except option A, formation of epinephrine from norepinephrine. Option B, formation of creatine. Option C, synthesis of serine. Option D, synthesis of melatonin from serotonin. And option E, formation of polyamine. S-adenosyl methionine is a methyl donor in all the reactions except basically, in which of the following reaction S-adenosylmethionine does not act as a methyl donor? Option A, formation of epinephrine from norepinephrine. Option B, formation of creatine. Option C, synthesis of serine. Option D, synthesis of melatonin from serotonin. And option E, formation of polyamine. You can write the answer in the chat box. Do you know the answer? Okay, you have answered option C, synthesis of serine. That is the correct answer. So apart from that reaction, in all these reactions like formation of epinephrine from norepinephrine or formation of creatine or synthesis of melatonin from serotonin. So we have already discussed epinephrine and melatonin serotonin synthesis in the previous class, right? So you had seen there was a methyl group transfer by S-adenosine methionine or abbreviated as SAM or SAM. So this methionine amino acid is very important in terms of methyl group donation. So wherever a methyl group is required, methionine is the amino acid that provides the methyl group in the form of S-adenosylmethionine. So this is just to again revise the reactions we had studied in the previous class. So from the norepinephrine to epinephrine synthesis, we know one methyl group is transferred to the amine group of norepinephrine, and we get epinephrine. And this methyl group transfer is done by phenyl ethanolamine N-methyl transferase. So whatever is the substrate in front of that, methyl transferase will be added. And since the methyl is transferred on the nitrogen, that's why N-methyl transferase. Okay, so phenyl ethanolamine N-methyl transferase. This is the enzyme. Methyl group donor is SAM. It will become S-adenosyl homocysteine and give the methyl group to norepinephrine to become epinephrine. Similarly, here melatonin synthesis from serotonin. So you can see we have serotonin here, and then the serotonin gets activated and become N-acetyl 5-hydroxytryptamine of N-acetyl serotonin basically. And this N-acetyl serotonin will then take up the methyl group from S-adenosylmethionine by the enzyme hydroxyindole O-methyl transferase. Now you see the methyl group is transferred at the OH part, so it is O-methyl transferase. Here we had N-methyl transferase. Here one more methyl transferase is involved in the serotonin synthesis itself. So we had 5-hydroxytryptophan from which serotonin was synthesized by decarboxylation. This 5-hydroxytryptophan can also become 5-methoxytryptophan by a methyl group transfer. That is also O-methyl transferase because the methyl group is transferred at the O position. Now, third reaction, that is the creatine synthesis. In this, basically glycine and arginine come together to form guanidoacetic acid, and this guanidoacetic acid is converted to creatine by a guanido acetate methyl transferase, and the methyl group is transferred here. You can see it's marked. So basically, S-adenosylmethionine can act as a methyl group donor for multiple reactions as mentioned. I hope that's clear. So this is just a list of methyl group acceptor and the final product. So I've shown you the creatine, epinephrine, and melatonin synthesis. Like that, there are multiple reactions where methyl group is donated to nicotinamide to become N-methyl nicotinamide, or methyl group is donated to ethanolamine to become choline. So like that, multiple reactions are there. We have tRNA also as a substrate where methyl group is given to tRNA to make methylated tRNA. Okay, so this is just a list of substrate that can take the methyl group from S-adenosyl methionine. So these are called transmethylation reaction because the methyl group is transferred from one substrate to another. So from the S-adenosyl methionine, the methyl group is transferred to another substrate or another methyl group acceptor. I hope that's clear. If you have any doubt, please let me know, okay? Moving on to the next question now. Homocystinuria type 1 is the uh, most often due to a deficiency of which enzyme? Homocystinuria type 1 is most often due to a deficiency of which enzyme? Option A, methionine synthase. Option B, cystathionine beta synthase. Option C, homocysteine methyl transferase. And option D, serine hydroxymethyl transferase. Homocystinuria type 1 is the most often due to a deficiency of which enzyme? Option A, methionine synthase. Option B, cystathionine beta synthase. Option C, homocysteine methyl transferase. And option D, serine hydroxymethyl transferase. You have answered option B, cystathionine beta synthase. That is the correct answer. So this is the metabolic pathway where this homocysteine you saw, it can get converted into methionine. It can also go towards the formation of cysteine by the help of serine. So the first product that forms with homocysteine and serine is cystathionine, and this cystathionine can break down into homoserine and cysteine. Okay. Now in this pathway, the cystathionine synthase is if the cystathionine synthase is deficient, there will be homocysteine accumulation, right? And that homocysteine accumulation will lead to homocysteine excretion in the urea. And the condition is called homocystinuria. Okay. So mostly it is autosomal recessive condition. Normal homocysteine level in the blood is 5 to 15 micromole per liter, but in diseases, it may be increased to 50 to 100 times. Okay. Moderate increase is seen in aged person. And then there are some acquired homocystinuria, which are due to vitamin B12 or B6 deficiency. So you see homocysteine breakdown into methionine requires vitamin B12. Uh, so if there's a vitamin B12 deficiency, that can also lead to homocystinuria. But that is an acquired condition. That is not a genetic condition. But then cystathione synthase deficiency, it's a genetic condition. It's an autosomal recessive condition. Okay. So it can, the acquired homocystinuria can get triggered by in the bodies of tobacco smokers, alcoholics, and in case of hypothyroidism also. And substantial increase is noticed in the congenital diseases. So this homocystinuria, it is generally asymptomatic in the initial years and symptoms develop later at the age of 3 to 5. Sorry. And the symptoms are abnormality of vision because of ectopia lentis, that is abnormal displacement of the eye lens into posterior chamber of the eye. Then second symptom is mental retardation. Third symptom is weak bone due to osteoporosis. And then there is increased vascular thrombosis and embolism leading to increased risk of coronary artery disease, cerebral stroke, etc. And the biochemical abnormalities is, I told you, homocysteine excretion in the urine and increased blood levels, blood level, basically homocysteine levels in the blood. So, and deficiency of cystathionine synthase can lead to accumulation of homocysteine in urine and blood, causing the condition called homocystinuria. Okay, I hope that's clear. Moving on to the next question.
Yes, ma'am.
Moving on to the next question now. Detection of homocysteine can be done in urine by option A, cyanogenin test. Option B, cyanide copper sulfate test. Option C, cyanide nitroprusside test. Option D, cyanide ferric chloride test. And option E, cyanide nitrogen test. Detection of homocysteine can be done in urine by cyanogenin test, cyanide copper sulfate test, cyanide nitroprusside test, cyanide ferric chloride test, and cyanide nitrogen test. You have given the answer option C, cyanide nitroprusside test. That is the correct answer. So I told you homocystinuria is basically accumulation of homocysteine and excretion of homocysteine in the urine. So the screening, obviously, we have to now detect whether a person is suffering from homocystinuria. We'll try to detect the homocysteine in the urine. So for detecting the homocysteine in the urine, cyanide nitroprusside test is done. This cyanide reduces the disulfide bond of homocysteine to produce homocysteine. So you see the spelling difference here. This is important. So this homocysteine, this is a dimeric form. So this homocysteine here, this is a dimeric form. It is written T-I-N-E, right? And if I'm writing T-E-I-N-E, that means it is a monomeric form. So you know there's a sulfhydryl group in the cysteine, right? And even in the homocysteine. So this sulfhydryl group has a tendency of forming a disulfide bond with another molecule of homocysteine. Okay, so two molecules of homocysteine connected together by this disulfide bond. It is written in the form of T-I-N-E. The spelling is T-I-N-E. And if it is a monomeric form, then it is written T-E-I-N-E. So this nomenclature is true for both cysteine and homocysteine. So if it is a dimeric form, now cyanide can reduce the dimeric form to produce the monomeric form. And the SH group of the homocysteine gives a red color with nitroprusside. So now this SH group will be free. Now this SH group will be free and it can react with nitroprusside to give a red color. Now if cysteine is present in urine, it is going to be misleading because cysteine also will have that SH group. So in that case, people along with cyanide nitroprusside test, they do silver nitroprusside test, which reacts with the SH group of cysteine to give positive test without reducing the disulfide bond. Okay, so homocysteine, the dimeric form, this homocysteine will not give the positive test with silver nitroprusside, but cysteine will. And if both cyanide nitroprusside and silver nitroprusside is positive, if cyanide nitroprusside is positive but silver nitroprusside is negative, then that means it is due to homocysteine in the urine, and that condition could be homocystinuria. Okay. And in the plasma, obviously, since cystathionine synthase activity, basically that enzyme is deficient. So you can confirm the estimation of that enzyme activity and detect the amounts of homocysteine in the plasma to detect homocystinuria. Okay. So first a urine screening is done. In case they see positive result in urine screening, then they move to the diagnosis in the plasma. Is that clear? Moving on to the next question then. Methionine, valine, and isoleucine are all catabolized to which TCA cycle intermediate? Methionine, valine, and isoleucine are all catabolized to which TCA cycle intermediate? Option A, fumarate. Option B, alpha ketoglutarate. Option C, succinyl coenzyme A. And option D, acetyl coenzyme A. Methionine, valine, and isoleucine are all catabolized to which TCA cycle intermediate? Fumarate, alpha ketoglutarate, succinyl coenzyme A, and acetyl coenzyme A. What could be the correct answer? You're saying succinyl coenzyme A. That is the correct answer. Very good. So, different amino acids have different roles in the metabolic uh, pathway. So ultimately, all the amino acids are either entering the glucogenic or the ketogenic phase, and some amino acids are there which are both glucogenic and ketogenic, but they will make some or the other, like they'll get converted into or catabolized into some or the other substrates that can enter the glycolysis or TCA cycle or gluconeogenesis pathway. So those are called glucogenic because they can ultimately go towards the formation of glucose. So methionine, it gets converted into homocysteine. We have discussed this earlier. From homocysteine, it gets converted into cysteine. From cysteine, it can get converted into alpha ketobutyrate by a transamination reaction. This alpha ketobutyrate can again get converted into propionyl coenzyme A, and propionyl coenzyme A can become succinyl coenzyme A, which can enter the TCA cycle. Okay, so succinyl coenzyme A, it's a substrate of TCA cycle. Now, similarly, valine can also get converted into propionyl coenzyme A, and isoleucine can also get converted into propionyl coenzyme A. So, and even threonine can get converted into alpha ketobutyrate and later enter the propionyl coenzyme and succinyl coenzyme pathway. Okay. So all these amino acids, methionine, isoleucine, valine, threonine, they all get converted into succinyl coenzyme that can enter the TCA cycle. Okay. Let's move to the next question. Cystathionine is due to a deficiency of option A, methionine synthase, option B, cystathionase, option C, homocysteinase, option D, arginase. Cystathionine is due to a deficiency of methionine synthase, cystathionase, homocysteinase, and arginase. You have answered option B, cystathionase. That is the correct answer. So we saw homocystinuria, right? Homocystinuria was due to cystathionine synthase deficiency. And because of that, cystathionine was not forming and homocysteine was getting accumulated. Now, cystathionine is caused due to accumulation of cystathionine. Okay, so cystathionine is excreted in the urine, and that's why it's called cystathionine. Now, cystathionine gets converted into homoserine and cysteine by the enzyme cystathionase. So I told you homocysteine along with serine forms cystathionine. Cystathionine gets broken down into homoserine and cysteine. Okay, and the enzyme that does this is cystathionase. Now, the deficiency of cystathionase will inhibit this reaction, so there will be no formation of cysteine or homoserine, and the cystathionine accumulation will start and eventually excreted in the urea, and it will be there in the blood circulation, and it is called cystathionine. So I've told you it's a cystathionase deficiency. It's a recessive inherited disorder, and the symptoms are severe mental retardation. The biochemical effect is excretion of cystathionine in the urine and blood, and the treatment is basically restrict methionine intake. So you know this pathway is coming from methionines, right? Methionine is getting converted into homocysteine. Homocysteine is getting converted into cystathionine. So if you restrict methionine intake in the diet, it can eventually control the symptoms. And also, since you are now restricting methionine diet, and then your cystathionase is not working, so there will be a deficiency of cysteine. So you have to give supplements of cysteine, PLP, folic acid. So whatever is the downstream of this reaction, those things have to be supplemented, and whatever is the upstream of this reaction has to be restricted in the diet. Okay. I hope you understood this. I'll move on to the next question then. Cysteine is catabolized to which glucogenic intermediate? Cysteine is catabolized to which glucogenic intermediate? Option A, fumarate. Option B, alpha-ketoglutarate. Option C, succinyl coenzyme A. And option D, pyruvate. Cysteine is catabolized to which glucogenic intermediate? Fumarate, alpha-ketoglutarate, succinyl coenzyme A, and pyruvate. Cysteine is catabolized to which glucogenic intermediate? Fumarate, alpha-ketoglutarate, succinyl coenzyme A, or pyruvate. So you saw for methionine, it was getting converted into propionyl coenzyme A and succinyl coenzyme A eventually. Similarly, how is cysteine catabolized? Where does it enter the glucogenic pathway? Fumarate, alpha-ketoglutarate, succinyl coenzyme A, or pyruvate? You've answered option D, pyruvate. That is the correct answer. So cysteine gets converted into pyruvate, and you know pyruvate enters the TCA cycle after getting converted into acetyl coenzyme A, and pyruvate can also get converted back to glucose by gluconeogenesis. So we have already covered the carbohydrate metabolism. So I hope that part is clear. So cysteine basically gets converted to pyruvate in two ways. Either a dioxygenase reacts with cysteine to form cysteine sulfonate, and this cysteine sulfonate is then undergoes a transamination reaction where the amino group is transferred to alpha keto acid. We have discussed the amino transferase or transamination reactions in the previous session. So I hope you understand this part. So cysteine sulfonate becomes beta sulfonopyruvate, and this beta sulfonopyruvate then gets desulfonated. The sulfide group is removed and it becomes pyruvate. Okay, so cysteine can get oxidized to form cysteine sulfonate. Cysteine sulfonate can then undergo transamination to form beta sulfonopyruvate and then undergo desulfonation to form pyruvate. Another pathway is, first step is transamination, where cysteine is transaminated to 3-mercaptopyruvate. So direct this amino group is without any oxidation. First amino group is transferred to alpha keto acid, and in turn we get another alpha keto acid which is 3-mercaptopyruvate. Then the sulfur transferase will come and take up this sulfur in the form of H2S, and the product that will be left with is pyruvate. Okay, so it can go via two pathways. Either it can undergo oxidation first, where the sulfur group will be oxidized, and then a transamination to form beta sulfonopyruvate, and later the sulfur group will be removed and we'll get pyruvate. Or the second pathway is cysteine can be directly transaminated by transaminase, and we will get 3-mercaptopyruvate, and later the sulfur group can be transferred in the form of H2S, and the product will be pyruvate. In both the cases, this pyruvate can enter the glucogenic pathway. I hope this is clear. We'll move on to the next question then. Which of the following enzymes is involved in the synthesis of glycine? Which of the following enzymes is involved in the synthesis of glycine? Option A, glycine decarboxylase. Option B, serine hydroxymethyl transferase. Option C, tyrosine aminotransferase. And option D, phenylalanine hydroxylase. Which of the following enzymes is involved in the synthesis of glycine? Glycine decarboxylase, serine hydroxymethyl transferase, tyrosine aminotransferase, phenylalanine hydroxylase. Which of the following enzymes is involved in the synthesis of glycine? Glycine decarboxylase, serine hydroxymethyl transferase, tyrosine aminotransferase, and phenylalanine hydroxylase. You've answered option B, that is serine hydroxymethyl transferase. That is the correct answer. So glycine can be synthesized from serine by this enzyme, serine hydroxymethyl transferase, and it is actually a reversible enzyme. So serine can also be formed from glycine by the same enzyme, and in this reaction, basically the beta carbon of serine. So this is the alpha carbon of serine. This is the beta carbon of serine. So beta carbon of serine is removed, and that enters in the 1-carbon pool with the help of tetrahydrofolic acid. So this is THFase tetrahydrofolic acid. It will take up this beta carbon and become NN-methylene tetrahydrofolic acid. And once this beta carbon is removed, we are left with the alpha amino acid, that is glycine. Okay, and the enzyme that is involved is serine hydroxymethyl transferase. So you see there is a methyl transfer from serine to tetrahydrofolic acid. Okay, so this type of methyl transfer is not S-adenosylmethionine dependent. This is direct methyl transfer from serine to tetrahydrofolic acid. Okay. And in the reverse reaction, when glycine is converted to serine, N-methylene tetrahydrofolic acid will donate the methylene group to the glycine to form serine. Okay, so hydroxymethyl group is transferred. This is a hydroxymethyl group, CH2OH. There's a OH attached to the methyl group. So it's called a hydroxymethyl group, and since the hydroxymethyl group is attached to serine, the enzyme is named as serine hydroxymethyl transferase. Okay. Let's see the next question now. Which enzyme synthesizes glycine from glyoxylate? Which enzyme synthesizes glycine from glyoxylate? Option A, glycine aminotransferase. Option B, glycine synthase. Option C, threonine aldolase. And option D, serine hydroxymethyl transferase. Which enzyme synthesizes glycine from glyoxylate? Glycine aminotransferase, glycine synthase, threonine aldolase, and serine hydroxymethyl transferase. You have answered option B, that is glycine synthase. The correct answer is actually glycine aminotransferase. So you see, glycine synthase actually synthesizes glycine from CO2 and NH3. So in the previous question, we saw the synthesis of glycine from serine. So glycine can actually get synthesized from multiple sources. Those are serine, threonine, carbon dioxide and ammonia directly, and glyoxylate. Okay, so there are 4 substrates for glycine. So glycine can get converted by serine, threonine, CO2 and NH3 directly, and then glyoxylate. Okay, so all these 4 molecules can act as a substrate for the glycine synthesis. So we saw glycine synthesis from serine by serine hydroxymethyl transferase. Glycine synthesis from threonine occurs by threonine aldolase. So it's a aldolation reaction. So the bond is broken from this part, and we get acetaldehyde and glycine. So threonine aldolase will break the bond, and we get 2 products, glycine and acetaldehyde. Okay. Then we have the next substrate, carbon dioxide and ammonia, that can that can synthesize glycine by the help of the enzyme glycine synthase, and it actually takes up the methylene group from N-methylene tetrahydrofolic acid. So remember, in case of serine hydroxymethyl transferase, tetrahydrofolic acid was taking the hydroxymethyl group to form N-methylene tetrahydrofolic acid. In this case, now the N tetrahydrofolic acid will give the methylene group, and the carbon dioxide and NH3 along with the methylene group will form glycine. And in this reaction, one NADH molecule is also utilized. Then we have the last reaction, that is glyoxylate. So glyoxylate gets converted into glycine. This is again a reversible reaction by the enzyme glycine aminotransferase. Glycine aminotransferase, by the name you can see it's a transamination reaction, right? So amine group from one amino acid, alanine, will be transferred to glyoxylate to form glycine, and alanine will get converted into pyruvate. So you remember the transamination reaction where amino group is transferred to from an amino acid to a keto acid, and the keto acid becomes one amino acid, and the amino acid gets converted into some other keto acid. And if you remember in the previous session also, the cofactor involved with the aminotransferase was PLP. And this enzyme is also known as alanine glyoxylate aminotransferase. So glycine aminotransferase or alanine glyoxylate aminotransferase, since the amine group transfer is happening between glyoxylate and alanine. Okay. So the enzyme that is responsible for the glycine synthesis from glyoxylate is glycine aminotransferase. Is this clear to you? Okay. Moving on to the next question. Creatine is synthesized from which combination of amino acids? Creatine is synthesized from which combination of amino acid? Option A, methionine, glycine, arginine. Option B, lysine, glycine, serine. Option C, methionine, alanine, proline. Option D, cysteine, glycine, lysine. Creatine is synthesized from which combination of amino acids? Option A, methionine, glycine, arginine. Option B, lysine, glycine, serine. Option C, methionine, alanine, proline. And option D, cysteine, glycine, lysine. You have answered option A, methionine, glycine, arginine. That is the correct answer. So I've shown you this creatine synthesis pathway in the first slide also. So glycine and arginine combines to form guanidoacetic acid and ornithine by the enzyme glycine arginine amidotransferase. So here amide group is getting transferred. Okay. So this is glycine and this is arginine. Okay. From arginine, this amide group is transferred and we get the compound guanidoacetic acid and ornithine. Okay. From guanidoacetic acid, then a methyl transfer reaction will occur, guanidoacetate methyl transferase, and the methyl group donor will be S-adenosylmethionine. And once the methyl group is transferred to this N, so it's the N-methyl transferase, we'll get the compound called creatine. Okay. So the first step of the reaction occurs in the mitochondria of kidney and pancreas. So this first step, glycine arginine amidotransferase, this occurs in the mitochondria and mitochondria of kidney and pancreas. It involves the transfer of guanidino group from arginine to glycine, catalyzed by glycine arginine amidotransferase to produce guanidoacetate that I have already explained. The second step is S-adenosylmethionine donates the methyl group to guanidoacetate to produce creatine. This methylation reaction occurs in liver. Okay. So the first step is occurring in mitochondria of kidney and pancreas. The second step is occurring in liver. Okay. So this is important for you to remember. In the creatine synthesis first of all occurs with the help of glycine, arginine, and methionine. Methionine is responsible for the methyl group transfer. Glycine and arginine together will form the guanidoacetic acid and ornithine. Guanidoacetic acid will get methylated to form the creatine in the second step. So the first step will occur in the mitochondria of kidney and pancreas, and the second step will occur in the liver. And finally, creatine will be synthesized. Is this clear?
Yes, ma'am, it's clear, ma'am.
Okay. Let's move to the next question then. Glutathione is synthesized from which combination of amino acids? Glutathione is synthesized from which combination of amino acids? Option A, methionine, glycine, arginine. Option B, lysine, glycine, serine. Option C, methionine, alanine, proline. Option D, glutamate, cysteine, glycine. Glutathione is synthesized from which combination of amino acids? Methionine, glycine, arginine, lysine, glycine, serine, methionine, alanine, proline, and glutamate, cysteine, glycine. You've answered option D. That is the correct answer. Glutamate, cysteine, glycine. So glutathione is basically a tripeptide called glutamyl cystinyl glycine. So it forms from these three amino acids. And this tripeptide, if you remember, in the previous sessions, I've told you how glutathione helps in the transport of amino acid across the membrane. So this is how glutathione is synthesized. So glutamate first combines with cysteine to form the glutamyl cysteine. So by the enzyme gamma glutamyl cysteine synthetase. Since it's a synthetase, there is a ATP hydrolysis involved that drives the energy for this reaction to go forward, and we get gamma glutamyl cysteine. This gamma glutamyl cysteine will then get combined with glycine by glutathione synthetase. Again, a synthetase, that means ATP hydrolysis is required, and then we'll get gamma glutamyl cysteanyl glycine, or it is called glutathione. It's written reduced because the cysteine part is in the reduced form. If it is in the uh, dimeric form, then it won't be reduced. So the free SH group is due to the reduced form of glutathione. And this glutathione helps in the transport of different amino acids across the membrane that I've explained in the previous session already. Glutamate becomes gamma glutamyl cysteine by the 1-cysteine incorporation by the enzyme gamma glutamyl cysteine synthetase. Then gamma glutamyl cysteine gets converted into glutathione by the enzyme glutathione synthetase when 1-glycine gets attached to this. I hope this is clear. We have discussed the glutathione role of glutathione in the previous session also. Moving on to the next question. Which disorder is characterized by the deficiency of the glycine cleavage system? Which disorder is characterized by a deficiency of the glycine cleavage system? Option A, alkaptonuria. Option B, maple syrup urine disease. Option C, glycogen storage disease. Option D, non-ketotic hyperglycemia. Which disorder is characterized by a deficiency of the glycine cleavage system? Option A, alkaptonuria. Option B, maple syrup urine disease. Option C, glycogen storage disease. And option D, non-ketotic hyperglycemia. Yes, option D is the correct answer. You are right. Non-ketotic hyperglycemia. So this is the glycine cleavage system where glycine is first decarboxylated by enzyme glycine decarboxylase. So the CO2 is removed, and what we are left with is a amino methyl group that gets converted into ammonia, and the enzyme involved is amino methyl transferase. So this glycine cleavage system is a multi-enzyme complex. It has four different proteins combined together, P, T, H, and L. So all of them have different roles associated with it. And glycine gets converted into ammonia and CO2. So this is a breakdown of glycine basically. And a defect in this glycine cleavage system can lead to non-ketotic hyperglycemia, also abbreviated as NKH. This is basically if the glycine is not cleaved, there will be accumulation of glycine, increased levels of glycine in the blood, urine, and CSF, that is the cerebrospinal fluid, and it can lead to severe mental retardation and seizures. Okay. So glycine, if it is not cleaved, if the cleavage system of glycine is defective, it can lead to accumulation of glycine in the blood, urine, and CSF, and that can lead to non-ketotic hyperglycemia, and the symptoms are severe mental retardation and seizures. I hope that's clear. Let's move to the next question then. Which non-essential amino acid is synthesized by the body from 3-phosphoglycerate? Which non-essential amino acid is synthesized by the body from 3-phosphoglycerate? Option A, proline. Option B, serine. Option C, histidine. And option D, lysine. Which non-essential amino acid is synthesized by the body from 3-phosphoglycerate? Option A, proline. Option B, serine. Option C, histidine. Option D, lysine. Which non-essential amino acid is synthesized by the body from 3-phosphoglycerate? Proline, serine, histidine, lysine. You have answered option B, that is serine. That is the correct answer. So serine can be synthesized from 3-phosphoglycerate. So this is the pathway how serine can be synthesized from 3-phosphoglycerate. Again, it can go through two pathways, one in cytosol and one in plastids. So we'll first see the first pathway. So in cytosol, which is called the glycerate pathway, non-phosphorylated pathway or non-phosphorylated glycerate pathway. Here, dephosphorylation occurs in the first step by the enzyme PGA phosphatase. Now, phosphate group has to be removed, so we need a phosphatase enzyme. So 3-phosphoglycerate will get converted into glycerate. So phosphoglycerate phosphatase will remove the phosphate group and become and the product will be glycerate. Now this glycerate will be uh, dehydrogenated to form hydroxypyruvate, and any dehydrogenase is associated with NADH, NADPH and dependent pathway. So glycerate dehydrogenase will convert glycerate into hydroxypyruvate, and in turn one NADP or NAD will be converted back to NADH or NADPH. Hydroxypyruvate will then get converted into serine by the enzyme serine glyoxylate uh, and serine pyruvate aminotransferase. So it's a aminotransferase or a transamination reaction where hydroxypyruvate is a keto acid, right? So glycine or alanine can uh, give the amino group to the hydroxypyruvate and get converted into glyoxylate or pyruvate, and this hydroxypyruvate will get converted into serine. Okay, so either serine glyoxylate or serine pyruvate aminotransferase can do this reaction. Now, in the second pathway, which is the phosphorylated pathway or phosphohydroxypyruvate pathway, in this step, the dephosphorylation doesn't happen in the first step. So in the cytosol, in the non-phosphorylated pathway, first step was dephosphorylation. We have to remove it since it's a non-phosphorylated pathway. We have to remove the phosphate first. So this was phosphatase acting on 3-phosphoglycerate. In the phosphorylated pathway, first 3-phosphoglycerate is dehydrogenated to form phosphohydroxypyruvate by the enzyme PGA dehydrogenase. This phosphohydroxypyruvate will undergo transamination. So glutamate will transfer the amino group, and glutamate will get converted into glutamate will get converted into oxoglutarate, and phosphohydroxypyruvate will get converted into phosphoserine. This phosphoserine will then dephosphorylated in the last step by phosphoserine phosphatase and form serine. So you see this is exactly opposite. So first in the cytosol, in the non-phosphorylated pathway, we had dephosphorylation first by a phosphatase enzyme, then dehydrogenation reaction by a dehydrogenase, and then aminotransfer reaction by a transaminase or aminotransferase. In the last state, in the phosphorylated pathway, it's first dehydrogenation and transamination is occurring, and then last step is dephosphorylation. Okay, so if the dephosphorylation is occurring in the first go, it is called the non-phosphorylated pathway, or if the dephosphorylation is occurring at the last step, it is called the phosphorylated pathway. Okay. So with whatever is the first metabolite that is forming after 3-phosphoglycerate, the pathway is named after that. So in this case, 3-phosphoglycerate is getting converted into glycerate by dephosphorylation. So it is called the non-phosphorylated pathway or also glycerate pathway. In this case, we have 3-phosphoglycerate getting converted into phosphohydroxypyruvate. So it is called the phosphorylated pathway and also known as phosphohydroxypyruvate pathway. Okay, so serine can be synthesized from both from 3-phosphoglycerate by both the pathways at different locations. I hope that's clear. Moving on to the next question. Which of the following glucogenic products is synthesized by serine? Option A, succinyl coenzyme A. Option B, fumarate. Option C, pyruvate. Option D, oxaloacetate. Which of the following glucogenic products is synthesized by serine? Succinyl coenzyme A, fumarate, pyruvate, oxaloacetate. Which of the following glucogenic product is synthesized by serine? Succinyl coenzyme A, fumarate, pyruvate, oxaloacetate. You are saying option C, pyruvate. That is the correct answer. So serine can be converted into pyruvate by serine dehydratase enzyme. So serine participates in the transamination reaction. It undergoes deamination to form pyruvate. Pyruvate can be converted to glucose or alanine as required. That we know that this is a gluconeogenesis pathway. So serine first, uh, it is dehydrated by the enzyme serine dehydratase. It is a PLP dependent enzyme. The H2O is removed from serine, and then the amino group is transferred. It is deaminated. So amino acid, this is deaminated to form pyruvate. Okay. This is how serine can get converted into pyruvate. So if you remember, glycine can also get converted into serine by the same enzyme we had studied, serine hydroxymethyl transferase. So glycine also enters the TCA cycle, the glucogenic uh, pathway, by getting converted into pyruvate. First, glycine gets converted into serine, and then serine enters into serine gets converted into pyruvate and enters the gluconeogenesis pathway. Is this clear? If you have any doubt till now, whatever is discussed, let me know. It's clear.
Yes, ma'am. It's clear, ma'am.
Okay. Okay. Moving to the next question then. The major enzyme for alanine metabolism, especially in gluconeogenesis, is alanine dehydrogenase, option B, alanine aminotransferase, option C, alanine transcarbamylase, and option D, alanine carbamoyl transferase. The major enzyme for alanine metabolism, especially in gluconeogenesis, is option A, alanine dehydrogenase, option B, alanine aminotransferase, option C, alanine transcarbamylase, and option D, alanine carbamoyl transferase. You have answered alanine aminotransferase, that is ALT. That is the correct answer. So we had discussed this aminotransferase, alanine aminotransferase and aspartate aminotransferase, ALT and AST, in the previous session also when we were reading about transamination reactions. So this alanine aminotransferase can actually catalyze the reaction where it can transfer the amino group from alanine to alpha ketoglutarate, and in turn we'll get glutamate from once alpha ketoglutarate takes up the amino group, it will become glutamate, and alanine, when it will give away the amino group, it will become pyruvate, and this pyruvate can obviously enter the gluconeogenesis pathway. So alanine can be formed by transamination of pyruvate. Alanine can form pyruvate by alanine can form pyruvate, which is a glucogenic substrate, and the enzyme involved is called the alanine aminotransferase, ALT. This enzyme is also called SGPT. Pyruvate, this reaction requires a pyridoxal phosphate, PLP. This is a PLP dependent pathway. And this ALT levels in blood is increased in case of liver diseases. So if any form of liver disease is present, so you must have seen, if you have ever gotten a blood test report, you might have seen ALT and AST levels mentioned, or SGPT, SGOT levels mentioned in the blood test report. So this ALT is basically that SGPT enzyme, and the increased level of ALT or increased level of SGPT in signals towards liver diseases. Okay. I hope that's clear. Let's see the next question now. Which important product is derived from the conversion of alanine and glucose alanine cycle? Which important product is derived from the conversion of alanine and glucose alanine cycle? Option A, pyruvate. Option B, acetyl coenzyme A. Option C, succinyl coenzyme A. Option D, oxaloacetate. Which important product is derived from the conversion of alanine in the glucose alanine cycle? Option A, pyruvate. Option B, acetyl coenzyme A. Option C, succinyl coenzyme A. Option D, oxaloacetate. You have answered option A, pyruvate. That is the correct answer. So, glucose alanine cycle is basically a pathway where uh, glucose is transferred from the muscles to the liver. So, glucose is converted to pyruvate in the muscles by the process glycolysis. We know that this pyruvate cannot directly be transferred from the muscles to the liver. So for that purpose, pyruvate is first converted into alanine by alanine aminotransferase. So this alanine aminotransferase is the muscle alanine aminotransferase, which converts pyruvate to alanine by taking up the amino group from glutamate and converting it to alpha ketoglutarate, and pyruvate becomes alanine. This alanine then comes in the blood and then enters the liver, where it is again converted into pyruvate by alanine aminotransferase present in the liver. So this alanine aminotransferase in liver is the ALT enzyme that we discussed in the previous slide. So this ALT, that's why I told you that the levels of ALT is very important in diagnosing some liver diseases, because it's a liver enzyme and it's a very important enzyme for pyruvate transfer across the muscle, from the muscle to liver. So alanine is then transferred to the liver, where alanine is converted to pyruvate, and then pyruvate can get converted into glucose by gluconeogenesis. So you know that gluconeogenesis occurs in liver, and this is how pyruvate is transferred from the muscle to the liver. So alanine is taken up by the liver from the peripheral tissue, particularly from the skeletal muscles, and it forms a major participant in the inter-organ transport of nitrogen. Okay, so transport of nitrogen occurs via alanine, because alanine is carrying that nitrogen, and then alanine will pass on that nitrogen to alpha ketoglutarate to form glutamate, and that glutamate can actually enter the urea cycle. So this we had discussed when we were discussing urea cycle as well. So all the nitrogen that is present in the peripheral tissue and muscle, they will be carried by this alanine, and then alanine will give the nitrogen to the glutamate. This glutamate in the liver can enter the urea cycle. And under conditions of starvation, obviously this glucose alanine cycle is important, because in case of starvation, all the carbon source go towards glucose formation so that more and more glycolysis can occur. So in case of starvation, blood glucose level is low. When blood glucose level is low, glycolysis cannot proceed. For that, first all the pyruvate is broken down into glucose again by gluconeogenesis, and all the other uh, glucogenic substrates are also pushed towards gluconeogenesis, and then the glucose is transported from the blood to the muscles where glycolysis can occur, and it can provide energy. So this is all about glucose alanine cycle, where alanine is the major participant in the transport of the pyruvate across the peripheral tissue. I hope that's clear. I hope you understood. Let's see the next question then. Which of the following amino acid is both glucogenic and ketogenic? Which of the following amino acid is both glucogenic and ketogenic? Glycine, valine, leucine, isoleucine. Which of the following amino acid is both glucogenic and ketogenic? Glycine, valine, leucine, and isoleucine. You are saying option C, leucine. That is the wrong answer. Actually, the correct answer is option D, isoleucine. So glycine, valine, and leucine all are glucogenic. Glycine and valine are glucogenic amino acid. Leucine is a ketogenic amino acid. Uh, Isoleucine is both glucogenic and ketogenic. So in the question I've asked, which of the amino acid is both glucogenic and ketogenic? So for that, the answer would be isoleucine. Isoleucine is both ketogenic and glucogenic. Isoleucine can enter in the metabolic pathway via acetyl coenzyme A, which is a ketogenic pathway. If you remember the ketone body synthesis from acetyl coenzyme A, we get the ketone body synthesized, ketone body produced, which was acetoacetate. And it can also enter the glucogenic pathway by entering the TCA cycle. Okay. Leucine can only enter the metabolic state by acetyl coenzyme A pathway, whereas glycine and valine are glucogenic substrates. Okay. So if it is asked that which of the following is both glucogenic and ketogenic, the right answer would be isoleucine. Leucine is only ketogenic. Valine is only glucogenic. Moving on to the next question. What is the first step in the catabolism of branched amino acid? What is the first step in the catabolism of branched chain amino acids? Option A, transamination. Option B, oxidative decarboxylation. Option C, dehydrogenation. Option D, phosphorylation. What is the first step in the catabolism of branched chain amino acid? Option A, transamination. Option B, oxidative decarboxylation. Option C, dehydrogenation. Option D, phosphorylation. What is the first step in the catabolism of branched chain amino acid? Transamination, oxidative decarboxylation, dehydrogenation, and phosphorylation. first step, okay? First step in the catabolism. You are answering option B, oxidative decarboxylation. That is the wrong answer actually. The first step is transamination. So branch chain amino acids are basically valine, isoleucine, and leucine. And the catabolism of these three amino acids, the first three steps are common in all three amino acids. And the first step is transamination. So transamination, as we know, removal of amino group from the amino acid to form a alpha keto acid. And it is catalyzed by a single vitamin B6 requiring enzyme, branched chain amino acid aminotransferase. So this branched chain amino acid aminotransferase, it transfers the amino group from valine, leucine, and isoleucine to form the respective uh, alpha keto acids. There are three isoenzymes actually of this branched chain amino acid transferase. So isoenzymes are basically the enzymes that can catalyze the same reaction but with different substrates. So these enzymes are similar in sequence and catalytic role, but they are present in different. So obviously there are three branch chain amino acids, right? So each enzyme out of these isoenzymes, each isoenzyme will be more specific towards one of the amino acids. So one will be more specific towards leucine, one will be more specific towards isoleucine, one will be more specific towards valine. And the reaction, the location of the reaction occurs in cytosol and mitochondria. Activity is higher in the muscles, and starvation induces this enzyme in the muscle, because during starvation we'll have to push the carbon containing compounds towards the glucose formation. So during starvation, all these amino acids are catabolized to enter some or the other glucogenic and ketogenic pathways so that it can produce energy in some way or the other. So this is the first step. The valine, leucine, and isoleucine, they are all transaminated by the enzyme branch chain amino acid transaminase to form the corresponding alpha keto acid. So valine gets converted into alpha keto isovalerate. Leucine gets converted into alpha keto isocaproate. And isoleucine is converted into alpha keto beta methyl valerate. Okay. So alpha keto isovaleric acid or isovalerate is the alpha keto acid of valine. So if you remove the amino group of valine, you will be left by alpha keto isovalerate. If you remove the amino group of leucine, you will be left by alpha keto isocaproic acid. And isoleucine will get converted into alpha keto beta methyl valeric acid or alpha keto beta methyl valerate. Okay. So first reaction is transamination, then comes the oxidative decarboxylation and dehydrogenation. Okay. So these three steps are common in the catabolism of valine, leucine, and isoleucine, the branch chain amino acid. The first three steps is common, and then the pathway bifurcates into three different steps. Valine uh, goes through a different pathway, leucine goes through a different one, and isoleucine goes through. I hope that's clear to you.
Yes, ma'am. It's clear, ma'am, now.
Okay. So next question is, which vitamin serves as a cofactor for the branch chain alpha keto acid dehydrogenase complex? Which vitamin serves as a cofactor for the branched chain alpha keto acid dehydrogenase complex? which vitamin serves as a cofactor for the branch chain alpha keto acid dehydrogenase complex? Option A, vitamin A. Option B, vitamin C. Option C, vitamin D. And option D, vitamin B1. Which vitamin serves as a cofactor for the branch chain alpha keto acid dehydrogenase complex? Option A, vitamin A. Option B, vitamin C. Option C, vitamin D. And option D, vitamin B1. You have answered option D, that is vitamin B1. That is the correct answer. So this is the second step of the catabolism of branch chain amino acids. So after transamination, the second reaction is oxidative decarboxylation. This reaction is catalyzed by the branch chain alpha keto acid dehydrogenase enzyme complex. And this complex is present in the inner mitochondrial membrane. Okay. And it is comparable in function to the pyruvate dehydrogenase complex. If you remember the pyruvate dehydrogenase complex reaction, uh, and it is this branch chain alpha keto acid dehydrogenase is similar to PDH complex, and it requires 5 coenzymes just like PDH: TPP, lipoamide, FAD, coenzyme A, and NAD plus. So TPP is thiamine pyrophosphate. So vitamin B1 is basically thiamine. So vitamin B1 is required for the uh, branch chain alpha keto acid dehydrogenase enzyme complex reaction, because it requires the cofactor or coenzyme TPP, thiamine pyrophosphate, that will be derived from vitamin B1. So if there's a deficiency of vitamin B1, this enzyme won't be working properly or won't be active. Alpha keto acid dehydrogenase catalyzes the oxidative decarboxylation of the keto acid to the corresponding acyl coenzyme A thioesters. Okay, acyl coenzyme A thioesters. This is a regulatory enzyme. Activity is increased by dephosphorylation. More active form occurs in the liver in well-fed state and muscle during starvation. So during starvation, in the muscles, it gets activated so that these amino acids could be catabolized and it can move towards the gluconeogenesis pathway. And in the liver, it is activated during the well-fed state so that these amino acids can be catabolized to form different glucogenic substrates. Okay. So you saw after the first reaction, the products were for valine, it was ketoisovalerate. For leucine, it is ketoisocaproate. For isoleucine, it was alpha keto beta methyl valerate. Okay. Now the second reaction, that is oxidative decarboxylation, it is carried out by branch chain alpha keto acid dehydrogenase. It's an enzyme complex, and it will give the corresponding acyl coenzyme thioesters. So for valine, after alpha keto isovalerate, it will get converted into isobutyryl coenzyme A. For leucine, alpha keto isocaproate will get converted into isovaleryl coenzyme A. For isoleucine, alpha keto beta methyl valerate will get converted into alpha methyl butyryl coenzyme A. Okay, so for valine, the product is isobutyryl coenzyme A. For leucine, the product is isovaleryl coenzyme A, and isoleucine, the product is alpha methyl butyryl coenzyme A. I hope that's clear.
Yes, ma'am.
And the next question is, What is the primary symptom associated with maple syrup urine disease? What is the primary symptom associated with maple syrup urine disease? Option A, mental retardation. Option B, elevated blood phenylalanine levels. Option C, sweet smelling urine. And option D, increased risk of hyperglycemia. What is the primary symptom associated with maple syrup urine disease? Option A, mental retardation. Option B, elevated blood phenylalanine levels. Option C, sweet smelling urine. And option D, increased risk of hyperglycemia. You are saying option C, sweet smelling urine. That is the correct answer. So maple syrup urine disease, it's an inborn error of metabolism. The urine of the affected individual smells like maple syrup or burnt sugar. That's why it's called maple syrup urine disease. So it has a sweet smell to the urine because of the uh, the enzyme that is defected in this case is branch chain alpha keto acid dehydrogenase. So this leads to the alpha keto acid accumulation in the urine that leads to the sweet smelling, sweet smell to the urine, like a maple syrup or burnt sugar, caramel like smell is there. Okay. Next question. Histidinemia is caused by option A, excess histidase. Option B, defective histidase. Option C, excess urocanase. And option D, defective glutamate dehydrogenase. Histidinemia is caused by option A, excess histidase. Option B, defective histidase. Option C, excess urocanase. And option D, defective glutamate dehydrogenase. Histidinemia. You are saying option B, defective histidase. That is the correct answer. So histidinemia, by the name you can understand, it's accumulation of histidine, increased level of histidine in blood. So the enzyme that is responsible for the catabolism of histidine, that is histidase, it converts histidine into urocanate, and deficiency or defect in this enzyme can lead to accumulation of histidine and eventually increased level of histidine in the blood. That will lead to histidinemia. Okay, this is the histidine catabolic pathway where histidine enters the glucogenic cycle by through alpha ketoglutarate. So histidine gets converted into urocanate by histidase. But this is a deamination reaction. Then urocanate by the enzyme urocanase gets converted into 4-imidazolone 5-propionate. This 4-imidazolone 5-propionate then gets hydrolyzed by the enzyme imidazolone propionate hydrolase to form N-formimino formiminoglutamate. This is also known as 5-glu. And this gets converted into glutamate and later to alpha ketoglutarate. This alpha ketoglutarate can enter the TCA cycle. Okay. So this is the histidine metabolic pathway, and histidine gets the first step of this pathway is the conversion of histidine to urocanate, which is by the enzyme histidase. And defect in this enzyme leads to accumulation of histidine and increased level of histidine in blood, leading to histidinemia. So it's an autosomal recessive disorder, and the deficiency of histidase leads to accumulation of histidine in the blood and body fluids and increased excretion of imidazole pyruvic acid in the urine. Clinical feature includes mental retardation and delayed speech development, low histidine diet may have some effect. So since histidine is getting accumulated, so if you uh, decrease the histidine in diet, that might help alleviate the symptoms. Okay. So histidine gets converted into imidazole pyruvic acid, which is secreted in the urine. Okay. Next question is, the main precursor of proline biosynthesis is. The main precursor of proline biosynthesis is option A, serine, option B, glutamate, option C, lysine, option D, alanine. The main precursor of proline biosynthesis is serine, glutamate, lysine, alanine. You are saying option B, glutamate. That is the correct answer. So glutamate is the precursor for proline synthesis. So glutamate and ornithine both can get converted into glutamate semialdehyde. This glutamate semialdehyde then gets uh, cyclized, spontaneous cyclization occurs, which can get converted into proline. So glutamate to glutamate semialdehyde is by the enzyme delta pyroline 5-carboxylate synthetase. And there's a ATP hydrolysis involved since it's a synthetase enzyme, and one NADPH oxidation is also involved. So delta pyroline 5-carboxylate synthetase. Then from glutamate semialdehyde, the spontaneous cyclization occurs, and then delta pyroline 5-carboxylate reductase enzyme converts the cyclized semialdehyde into proline. Okay, so two enzymes are involved here, pyroline 5-carboxylate reductase and L-proline dehydrogenase. Since there's a dehydrogenase involved, obviously there's a NADPH involvement. Okay, so glutamate is the precursor of proline. I hope that's clear. Moving on to the next question. Which amino acid acts as a precursor for carnitine synthesis? Which amino acid acts as a precursor for carnitine synthesis? Option A, histidine. Option B, proline. Option C, arginine. Option D, lysine. Option E, glutamate. Which amino acid acts as a precursor for carnitine synthesis? Histidine, proline, arginine, lysine, glutamate. Which amino acid acts as a precursor for carnitine synthesis? Histidine, proline, arginine, lysine, or glutamate? You are saying option D, lysine. That is the correct answer. So carnitine synthesis, this is the carnitine synthesis pathway. So trimethyl lysine serves as the precursor for the synthesis of carnitine. So lysine, when protein is degraded, generally lysine is present in the protein in the form of methylated, in the methylated form. And when protein degradation occurs, trimethyl lysine is released, and this trimethyl lysine serves as the precursor for carnitine. So first step in the pathway is trimethyl lysine dioxygenase, where oxidation of trimethyl lysine occurs to form 3-hydroxy trimethyl lysine. Then the second step is HTML, that is 3-hydroxy and trimethyl lysine, HTML, aldolase, where a glycine is released from this molecule, and the product that we get is 4N trimethyl aminobutyraldehyde, or it is also abbreviated as TMABA. And then TMBA dehydrogenase acts on this to convert into 4N trimethyl aminobutyrate. And the last step is gamma butyrobutane dioxygenase, which acts on, so 4N trimethyl aminobutyrate is also known as gamma butyrobutane, and gamma butyrobutane dioxygenase will oxidize this into carnitine. Okay, both the dioxygenase step is connected with alpha ketoglutarate to succinate formation. Okay, so trimethyl lysine serves as the precursor for carnitine, as I've already explained. Carnitine is involved in the transport of fatty acid to mitochondria for oxidation. And the synthesis of carnitine is a four-step reaction involving oxidation, splitting of glycine residue, dehydrogenation, and finally again oxidation. So first oxidation is by dioxygenase, where alpha ketoglutarate is reduced to succinate, and TML is oxidized to HTML. This HTML is then uh, it's a splitting reaction where glycine is removed, and the product that we are getting is TMABA. Then the third step is dehydrogenation, where this is getting converted to gamma butyrobutane. Gamma butyrobutane is then again oxidized to form carnitine, and again one molecule of alpha ketoglutarate is converted to succinate. Is this clear? The four steps of carnitine synthesis. Let me know if you have any doubt. Okay, moving to the next question then. A defect in the arginase leads to elevated levels of which amino acid? A defect in the arginase leads to the elevated levels of which amino acid? Option A, ornithine. Option B, arginine. Option C, glutamine. Option D, glutamate. A defect in the arginase leads to elevated levels of which amino acid? Option A, ornithine. Option B, arginine. Option C, glutamine. Option D, glutamate. You are answering option B, arginine. That is the correct answer. So arginase is the enzyme that is involved in the catabolism of arginine into ornithine. So arginine gets converted into ornithine by the enzyme arginase. So this you have seen in the urea cycle as well, this reaction. And the enzyme arginase, defect in the enzyme arginase will lead to accumulation of arginine. So like in case of histidine, we had histidase, and defect in histidase was leading to accumulation of histidine. Similarly, defect in arginase will lead to accumulation of arginine. Okay. So this was the last question of today's session. So let me know if you have any doubt in any of the questions that we have discussed today.
Ma'am, so nitroprusside test for homocystinureas. There's
Mhm.
another test called silver nitroprusside
Yes.
test. What is the difference, and what is the significance of these two tests in
Okay.
diagnosing homocystinuria?
Yeah. Yeah. So there are two different tests: cyanide nitroprusside test and silver nitroprusside test. Okay. So cyanide nitroprusside test, we have a cyanide. along with the nitroprusside. Okay. And for silver nitroprusside, we have the silver along with the nitroprusside. Okay. So now cyanide has the ability to break down the disulfide bonds. So homocysteine, since it has a sulfhydryl groups have a tendency to form disulfide bond. Okay. So if I have a structure somewhere. uh, So this is homocysteine, right? This is homocysteine. You see this SH group. So this is written TEINE, right? This is the monomeric form of homocysteine. But since this SH group is very reactive, they generally form dimer and they form homocysteine, TINE. Okay. And basically difference between this homocysteine and this homocysteine is the dimeric form. So this is the dimeric form. What do you mean by dimer? Basically, the this whole group I'm writing it as R. This whole part I'm writing it as R. So we have RSH, right? This is the monomeric form. This can form a dimer with another homocysteine molecule, and this will be homocysteine, TINE, and this will be TEINE. Okay. So this is the difference between homocysteine and homocysteine. Now, what was I saying? Cyanide nitroprusside test. So cyanide can break this disulfide bond. So cyanide can convert this homocysteine to homocysteine. Okay. So in the presence of cyanide, this disulfide bond will be reduced and we'll get the reduced form of homocysteine. That is, this free SH group will be there. Okay. Now, so this free SH group will be there, and this free SH group can react with the just wait for a second. Yeah. So cyanide can reduce the disulfide bond and convert it into the monomeric form and with the free SH group, free sulfhydryl group. And this free sulfhydryl group can react with the nitroprusside to give a red color. Okay. Now, in the urine, we have cysteine present as well. Okay. So that cysteine also has a free sulfhydryl group, right? So this cysteine can now react with that nitroprusside and give a red color. But how will we know whether the red color that we are seeing is due to the homocysteine or cysteine? Okay. Or homocysteine or cysteine? Okay. So now in that case, what people do is they combine this uh, cyanide nitroprusside test with the silver nitroprusside test. Now, silver nitroprusside test can react with the SH group of cysteine without reducing the disulfide bond. So, silver cannot reduce the disulfide bond. So, if homocysteine is present in this form, it won't react with the nitroprusside when treated with silver nitroprusside, and it won't give a red color. So a positive cyanide nitroprusside test where the nitroprusside is turning red, and a negative silver nitroprusside test where the nitroprusside is not turning red, that is suggestive of homocysteinuria. If the cyanide nitroprusside test is positive, that means it can either have homocysteine or cysteine. But if the negative silver nitroprusside test is also positive, that means it is showing positive result for cysteine. But silver nitroprusside test negative means homocysteine is present. That's why we are seeing a positive cyanide nitroprusside test. Okay. So cyanide nitroprusside test can give positive results. Positive result is the red color. Okay. So positive result for both homocysteine. Just a second. It can give a positive result for both homocysteine and the monomeric form of cysteine that is already present in urine. So if we do just this test, we won't understand whether we are actually having homocysteine in the urine or cysteine in the urine. So if we combine with the silver nitroprusside test, if the silver nitroprusside test is positive, that means we have cysteine in the urine, which is normal. If the silver nitroprusside test is negative, then we have homocysteine, because silver nitroprusside will not react with homocysteine because it cannot break the disulfide bond. So it won't react with, silver won't react with homocysteine, and then the free sulfhydryl group will not be present. So there will be no color change in case of uh, nit in case of silver nitroprusside. Okay. Is this clear now?
Okay, ma'am. Yes, ma'am. Yes, thank you, ma'am.
Yeah. So any other question you have, you can ask. If you don't understand anything that I have explained, you can ask again. I can explain again. Is it clear?
Yes, ma'am. It's clear, ma'am.
Okay. So if there's no other doubt, then I think I can end this session. Thank you for joining. I'll take one more session tomorrow. So see you tomorrow. Okay, tomorrow I'll discuss the 10th week's problem solving,
Okay,
10th
ma'am.
week's assignment. Okay.
Okay, ma'am.
Okay. Thank you. Bye bye.